This video presents overview of the molecular biohistory. That is, how molecules of intermolecular bonding brought birth of creature is explained. The first stage of molecular biohistory is bubble in liquid water. Gas in the bubble is enclosed by cage-shaped organization of water molecules formed by hydrogen bond. Although intermolecular interactions are individually weak, those are significant for the organization. The bubble and cage-shaped organization of water molecules lost the life together by intermolecular bonding mechanism. Here, the linked atoms and molecules are replaced by thermal movement. The electronic structure adapts to the surrounding environment. These reactions will affect the organization of molecules, but the intermolecular interactions are not chemical reactions. The molecule of water in liquid state links three-dimensionally by an hydrogen bond. Molecular structure of liquid water has been reported that there exists ice-like microstructures among the distorted hydrogen-bonded water molecules. There is the spiral structure as one of various structures of intermolecular bonding state of water molecules. Such structures are repeatedly generated and disappeared. The lifetime of clustered configuration has been estimated as time scale of picosecond. CO2 molecule induces forming of structured water. Molecules of water in liquid water will be rearranged under the influence of neighboring molecule of the other kind. Carbon dioxide is soluble in water. But carbonated water, 98.3% of the CO2 is in linear molecules. There is a through-hole in the spiral structure of water. The linear molecule of CO2 will be incorporated in the through-hole. So, bubbles of CO2 emerge at shock from outside in carbonated water. Unit of water molecule deforms from tetrahedron, because oxygen atom of water molecule connects with two hydrogen atoms by covalent bond. Deformed tetrahedral unit of water molecule connects each other by hydrogen bond. The HOH angle of covalent bond of water molecule is 104.5 degrees. Although the hydrogen bond will be the strongest, when OHO angle is 180 degrees, the angle of hydrogen bond is refracted by deformed tetrahedron. When one of the bond is vertical to the interface, the other bond of hydrogen atom aligned at different angle to the surface. The molecules of water can be configured to spiral structure. Thermal vibration of water molecules in liquid water will be configured to systematic movements, by keeping the spiral structure. The direction of refraction angle on the hydrogen bond OHO is changed by turns along each three kinds of electric axes. This thermal vibration causes a breathing motion, by accompanying stretch of the space. The intermittent movement will support synthesis reaction. The swirling motion of spiral structure will be induced by a macroscopic movement. The rotation of spiral structure of molecules moves the neighboring molecules. The three-dimensional conformation of filamentous proteins takes place immediately after emitted from the molecular organization. The protein cannot be produced at a time. Polymer of nucleic acid such as deoxyribonucleic acid or ribonucleic acid, that controls the synthesis of proteins has a spiral structure. So, the synthesis of protein is proceeded with the help of the respiratory movement of the spiral structure of water. Various size of respiratory movements can be observed in thawing the ice of carbonated water with containing glucose. Observations of the phenomena caused by behavior of spiral structure in water. Followings were confirmed by slow-motion replay of movie photographed at 30 frames per second. 1. Rising bubble of carbon dioxide in carbonated water outputs white area from the bottom. Concurrently sudden move of bubble takes place. 2. Sudden widening of water in carbonated water was generated a coalesce of bubbles. The widening of water is caused by existence of carbon dioxide. 3. The white area of water in carbonated water behaves as if it prevents from separation of carbon dioxide bubbles. These phenomena can be understood as follows. Linear molecule of CO2 may configure spiral structured water, by placing in through hole of the spiral. The spiral structure is formed at vertical direction to the plane of bubble. Here, Electric polarization of H2O is aligned along 120 degrees directionally arranged electric axis of water. When the spiral structure is formed between two bubbles, it will condense the volume. The energy of the condensed structure is low. So, two bubbles are attracted by the spiral structure. In case of transport of CO2 along through hole of spiral structure, vibrational thermal motion of water molecule turns to one-directional rotation. So, instantaneous widening of the water around the bubbles is observed accompanying with the quick movement. At the time when C had been born, principal gas in the atmosphere of Earth was CO2, and banded iron layers were formed as the result of frequent eruptions of submarine volcanoes. Following experiments were carried out to reproduce such phenomena. Fine powder of iron is mixed with carbonated water. Bubble of CO2 rise to surface of water. Fine iron powders attached to membrane of the bubble and those were oxidized. Robust bubbles of CO2 are formed after several hours. Eventually, floating matter and membrane on wall of glass container were remained. This is explained as follows. Carbonate ions are contained a little in carbonated water. Carbon atom that is surrounded by 3 oxygen atom in the isolated carbonate ions is exchanged by iron atom over time. Produced iron oxide is deposited at the bottom of water, and free carbon makes the membrane robust. On the other hand, deoxygenation reaction of H2O, due to oxidation of iron generates free hydrogen and iron hydroxide. Here, iron hydroxide dropped down to the bottom of water. Membrane of bubbles that is formed in carbonated water mixed with powder of iron. Fine particle of iron sticks to the membrane surface of CO2 bubbles at the first stage. In the course of time, the iron particle is oxidized and it is deposited to the bottom of water. Free carbon is produced by deoxygenation of CO2, that is placed in through holes of spiral structure, where carbohydrate is produced from free carbon and water molecule. Hydrocarbon is produced from free carbon and free hydrogen. Fatty acid is produced by exchanging a carboxyl group COOH to the hydrogen atom of end of hydrocarbon chain. Processing of the synthesis is completed by connection of glycerol. Phosphorylation of the membrane made of hydrocarbon. Hydrocarbon is hydrophobic. So, linear molecules of hydrocarbon are gathered and form a membrane. Electric potential on inside of the membrane becomes positive by the deoxygenation. So, negative ion of phosphoric acid connects to the outside of the membrane. It prevents invasion of electron to inside of membrane. Since the size of phosphoric acid is large, it is not able to penetrate into inside the membrane of hydrocarbons. On the other hand, oxygen atoms in phosphoric acid are arranged as a tetrahedron, and it will make linkage with water molecules. So, the phosphoric acid is able to mix with water in any proportion. Here, the electronegativity of phosphorus is nearly the same value with hydrogen. In the cell membrane, 2 fatty acids and 2 hydroxyl group of glycerol are joined by dehydration. Remaining 1 hydroxyl group and phosphoric acid are joined by dehydration. Biochemical reaction consists of dehydration synthesis and hydrolysis. Dehydration synthesis and hydrolysis on polypeptides, polynucleotides, and polysaccharides. Synthesis of nucleic acids based on structured water molecules. Carbohydrate includes monosaccharide glucose and polysaccharide cellulose. The pentose sugar fits to structure of water molecules. Every carbon atom of pentose sugar is able to match with the position of oxygen atoms of water. Because HOH angle on water molecules is 104.5 degrees, close to the inner angles of pentagons 108 degrees. So, glucose 6-phosphate is produced by phosphorylation of glucose on the 6th carbon. G6P cannot easily cross the cell membrane by the charge of phosphate group. Nucleic acids, which include deoxyribonucleic acid and ribonucleic acid, are made from nucleotide. Each ribonucleic acid nucleotide has a 5-carbon sugar, a phosphate group, and a nitrogenous base. Adenosine triphosphate consists of an adenine and ribose and triphosphate. Adenosine triphosphate is highly soluble in water. Adenosine triphosphate provides the phosphoric acid that functions as catalyst of biochemical reactions. Phosphoric acid functions as catalyst of biochemical reaction. Adenosine triphosphate, as well as ribonucleic acid and phosphate lipid membrane are composed of a pentose with phosphate. Adenosine triphosphate is highly soluble in water. Adenosine triphosphate releases phosphate through hydrolysis. Adenosine triphosphate is reproduced from the adenosine diphosphate that couples with H3PO4 by the dehydration that accompanies with absorbing energy. There is the mechanism that H2PO4 is exchanged with OH of amino acid residues, and it changes the three-dimensional structure of the protein. In addition, following mechanism can be existed, i.e. H2PO4 ion of adenosine triphosphate work as a catalyst on hydrolysis of the molecule on the membrane related to respiratory movement of the lipid membrane. Since the size H2PO4 ion is large, OH ion of molecule on the membrane will be exchanged to H2PO4 ion under the stress of expansion. The negative charge of phosphate ion collects protons. H2PO4 ion state in the membrane will be exchanged by OH ion at the time when the membrane is compressed. OH ion and collected proton of H are carried out hydrolysis reaction. Synthesis of protein and formation of the memory. Thermal motion of amino acid will be suppressed if it adheres to cell membrane. So amino acids are connected by peptide bonds in the vicinity of membrane. The polypeptide forms protein secondary structure, such as alpha helical cylindrical form or sheet form of beta structure by hydrogen bond between N, H and C, O. Moreover, alpha structure and beta structure are able to connect. Side chain of amino acid sticks out on the outside in such structure. There are intermolecular interactions between distribution of those molecules and molecules in the vicinity. Such organized molecules equipped with various functions. Protein is synthesized by a chain of catalytic reactions. On the other hand, synthesis of nucleic acids progresses intermittently by touching the cell membrane through movement of contraction and expansion. So, the relationships between the chain of reactions for a protein synthesis and sequence of nucleic acid are structured. The protein can be reproduced by calling the chain reaction of protein synthesis by nucleic acid sequences. Formation of network of information processing by chain reaction. The transference of reaction on a control is one direction. Bidirectional reaction is not available in a control system. So, the element that controls must be different from the element that is controlled. A control system must be configured by two different substances. Element of nucleic acid is different from element of protein. But both syntheses are progressed by catalysts. Activity of protein synthesis is catalyzed by nucleic acid. On the other hand, synthesis of nucleic acid is catalyzed by enzyme of protein. The reaction of intermolecular bonding takes place by replacement of molecules. Such reaction is carried out within the range of thermal motion. The chain reaction proceeds along one direction, due to the refractory period. The activities of life are controlled by chain reactions. The transmission of information by chain reaction is similar mechanism to neural network. The stimuli are sent from many leased lines. The control by use of many leased lines is similar to a puppet. Division of labor to synthesize proteins. The replication of protein is carried out through the division of labor that is composed of three parts. Three kinds of parts were divided from one primitive system to synthesize the protein. The division of labor makes possible to reproduce long proteins. The ribonucleic acid that carries one amino acid T-ribonucleic acid attaches one amino acids and corresponding ribonucleic acid. On the other hand, M-ribonucleic acid specify the amino acid sequence of one protein. The ribosome forms a protein chain by using an M-ribonucleic acid molecule and T-ribonucleic acid molecules. The system had evolved by natural selection keeping to common code, to specify the amino acid sequence in the M-ribonucleic acid and T-ribonucleic acid. Eventually, 3 nucleotide sets called codons are used to assign an amino acid. Origin of the deoxyribonucleic acid that stores genetic information. Since lifetime of ribonucleic acid is short, replication of the ribonucleic acid is needed. Primitive facility to replicate ribonucleic acid is the facility that produced original ribonucleic acid. But the facility will be large-scale if ribonucleic acid becomes long. Deoxyribonucleic acid is a stable molecule owing to double helical structure, and its nucleotide sequence may correspond to ribonucleic acid. But it is difficult to generate the deoxyribonucleic acid that corresponds by random processing. At the time where primitive deoxyribonucleic acid was begun to use for the memory of genetic information, probably it was possible to transfer the nucleic acid sequence from ribonucleic acid to deoxyribonucleic acids. That is, the nucleotide sequence of useful ribonucleic acid was transcribed to deoxyribonucleic acid. After that, the ribonucleic acid was replicated by using deoxyribonucleic acid that had been transcribed. Replication of deoxyribonucleic acid is carried out by following movements. Double lines of double helix become partially free by rotation in the opposite direction of spiral. So, processing of base sequence on the part of deoxyribonucleic acid is carried out. Then, the next part will be processed. Transmission of genetic information by the central dogma. The system that genetic information is transferred from ribonucleic acid to deoxyribonucleic acid creates long deoxyribonucleic acid molecules. But it causes a new problem because nucleotide sequences of ribonucleic acid will be easily changed. Many have blindly transferred deoxyribonucleic acid becomes obstacle of survive. After the state where the system equipped necessary deoxyribonucleic acids, mechanism to transfer nucleotide sequence of ribonucleic acid to deoxyribonucleic acid became extinct. Eventually, transfer of genetic information became one direction from deoxyribonucleic acid to ribonucleic acid. The flow of genetic information is explained by the central dogma as follows. Deoxyribonucleic acid is replicated into more deoxyribonucleic acid. Deoxyribonucleic acid is transcribed into ribonucleic acid. Ribonucleic acid is translated into protein. But transference of genetic information from protein to nucleic acid has been forbidden. The feature that had functioned in the past will be removed, if it becomes to harm on it. Summary. History that gave birth to life was explained as dialectical activities in ecosystem of molecules. Living creature was configured of different types of molecules by intermolecular bonds. Neighboring atoms in a creature are exchanged by thermal motion of liquid state of molecules. Water molecules in carbonated water will form the spiral structure. The spiral structure is able to make breathing movements and the turning motions. Activated carbon atoms are produced from carbonated water by oxygenation of iron atom. Activated hydrogen atoms are produced by oxygenation of iron atom in the water. Activities of life are supported by activities of protein. The protein is reproduced by calling the chain reaction of protein synthesis by nucleic acid sequences, after relationships between the chain of reactions for a protein synthesis and sequence of nucleic acid are structured. After the state, where the system equipped necessary deoxyribonucleic acid mechanism to transfer nucleotide sequence of ribonucleic acid to deoxyribonucleic acid became extinct. The feature that had functioned in the past will be removed, if it becomes to harm on it. Thank you very much for your attention.